Let's try this again. Apparently my first recording had some issues. I'm not sure what caused it, so I might do a few shorter videos just to make sure basically that it's working and I don't have to keep redoing these. So sorry. Okay, let's go ahead and get started. Epsilonproteobacteria. Our examples are Helicobacter and Campylobacter. Helicobacter pylori is the species that I've chosen for Helicobacter. There are other species, but pylori is the one of medical concern, or of most medical concern. About 50% of the entire global population will actually have an H. pylori infection, and only about 15% of those people infected will develop complications from this infection. Complications can include gastric or duodenal ulcers. Remember, duodenal refers to the very first part of your intestine immediately after your stomach. And of those 15% that do develop an ulcer, about 1 to 3% will actually develop stomach cancer. And actually, I said that a little bit wrong. Of the 50% that are infected, only 1 to 3% will develop stomach cancer, whereas 15% will develop some type of ulcer. Having the ulcer doesn't mean that you will develop stomach cancer. Also, not having an ulcer doesn't mean that you're not going to develop the cancer, right? So they're independent of each other. You certainly can have an ulcer and eventually have cancer, but like I said, it's not necessarily like a natural progression. But what we do see with the stomach is that repeated irritation, repeated damage does tend to increase the likelihood of cancer. It does seem to eventually lead to cancer for some people. We also have found that people with blood type O seem to be more prone to these complications of getting an ulcer and or cancer. H. pylori, it has these flagella on both ends, notice, and it's also this spiral shape, so it's really good at getting into the mucus layer, which lines it sits right on top of the lining of the stomach to protect it from stomach acid. So when that mucus layer gets disrupted, then the acid can come in contact with the actual epithelium, irritate it, and cause an ulcer. Another interesting fact about H. pylori is that it is able to convert the stomach acid with a special enzyme that it has called urease, and then that produces ammonia, and that is an alkaline substance. So it actually makes a little miniature climate around itself that is not acidic. And so that's a very special property of it as well. And it is microaerophilic, and you maybe don't know what that means. So micro, small, and air, philic, liking. So it likes very little amounts of oxygen. Okay, we're going to see that same property for Campylobacter, and in fact, they look pretty similar. So Campylobacter is the number one cause of diarrhea in the United States. It is gastroenteritis, and for some people, the infection is severe enough or causes such a severe response from the immune system that you can have really, really bad cramping. You can even have dysentery, right? Bloody diarrhea. And this, I mentioned it looks similar to Helicobacter, but notice It only has a single flagellum at one end. This treatment is typically just rehydration to treat the diarrhea, but if it's longer than a week of diarrhea, then a physician would prescribe an antibiotic, such as azithromycin. Something to note is that because it's a microaerophilic substance, it prefers the environment of the gut. It actually doesn't replicate in the food. It's just like the pause button effect, right? So it's the food's contaminated. There's lots of it. You ingest it, and once it gets into your gut, into your small intestine, it then it starts to replicate very rapidly and colonize the small intestine, and that is why the response is diarrhea, right? Because that's the best way to flush it out, to stop it from colonizing and to get rid of it. Okay. So Campylobacter, different species, is all over chicken. Like almost all chicken is contaminated with Campylobacter, and even 60% of cows excrete it in their feces and also in their milk. So this is why pasteurizing your milk is so important, because cows, they can secrete all sorts of different bacteria in their milk, such as Campylobacter. They also have bacteria on their skin that can get into the milk from the milking process. So it's very dangerous, very, very dangerous to drink unpasteurized milk. You're putting yourself at risk of a lot of different diseases. Pasteurization is not a chemical process. You're just heating the milk up to a high temperature, just high enough and long enough to kill the pathogens, which are more susceptible to that high heat. You don't kill all the bacteria. There's bacteria that are not harmful, that eventually will spoil the milk, right? But by pasteurizing it with that short high temperature burst is enough to kill the pathogens, and then you don't get sick when you drink the milk. You actually probably wouldn't get sick if you drink sour milk. It just obviously would taste terrible. So there you go. Undercooked chicken seems like a weird, like, who's undercooking their chicken? I think everyone knows not to do that. But in the UK, chicken and like chicken liver and things are used to make pate, which actually would be undercooked in that situation. You also have the issue of cross-contamination with chicken. Your cutting board, your knives, and your hands will all be contaminated with Campylobacter and possibly Salmonella and things. So you have to be careful when you're cooking chicken that once you get the chicken how you want it to put it into the pan or the oven, that you wash your hands, you wash anything that it came in contact with before you do anything else, okay? Especially if you're going to be having a salad, I recommend having a different cutting board that you use for things that will be raw, and then a special cutting board that's for meat that you're going to obviously then cook, right? That way you're reducing your chance of cross-contamination that way as well. Okay, so of course people who are working with chickens directly or with cattle directly need to also be careful to wash their hands after working with anything to do with the cattle. There's about over 1 million cases every year in the United States just from Campylobacter infection. So it's very real. But usually it's fine. 1 week resolves itself. But for some people, it does become more severe. It can last up to 3 weeks. As I mentioned, after 1 week, you would go to the doctor and get those antibiotics to help you out. The incubation period is pretty short. Once you ingest it, usually 2 days, 1 to 2 days is all it usually takes to get the start getting the symptoms, but it can take up to 5 days. A rare complication would be a temporary paralysis due to a temporary autoimmune attack. If you want to read more about that, you can check it out in your textbook. Okay, so now we're going to look at the Firmicutes. These are gram-positive. When we say low GC, remember we're talking about DNA. Remember the base pairs of DNA, A, T, C, G. And so these group of gram-positive bacteria have more A, T base pairs in their DNA and fewer GC. And so this is actually a nice quick way to see what you're dealing with. If you do just a quick little DNA test, you isolate the DNA, and then you can do things like weighing it. You can do like a mass spec, for example, and so there are different ways to figure out what the composition is, and it's relatively simple and fast. Okay, so let's get started. Remember dichotomous keys? It's always a either or question. So starting out at the top, it's saying low GC gram-positive. So assuming that you've already gotten to this point and the bacteria that you're working with is a gram-positive low GC bacteria, then the next step is to determine, does it have a cell wall or not? If it does not have a cell wall, it's a mycoplasma. We very briefly mentioned that in chapter 4, and we're not really going to look at any examples of that, but they are the tiniest cell that we know of, so it's kind of interesting. On the right would be everything that we're going to actually look at. So they have cell walls, and they either produce endospores or they do not. Remember, endospores are those highly resistant and dormant structures. They bacteria are just, again, that pause button has been pushed. They're just surviving. Okay, once they're in a good environment, they will germinate and return to the vegetative state. But while it's an endospore, it is dormant. It is not reproducing. It is not actively undergoing any metabolic function. Okay. So if it produces endospores, then we want to know what kind of oxygen requirements it has. Is it completely anaerobic, or is it facultatively anaerobic? Or maybe it's actually aerobic. So aerobic, think about aerobic exercise. You're breathing a lot, so you do utilize oxygen. Anaerobic would mean without oxygen. If it's facultative, it means it can do both. It can utilize oxygen when it's available, and it can survive without oxygen when it's not available. Okay. And so we'll look at Clostridia that are anaerobic, and they have endospores. And then we'll also briefly look at Lactobacillus and a couple other examples. Okay. So Clostridium species, we're actually going to look at 4, not 5. Please correct that. I removed the 5th one because it doesn't impact humans. It impacts cattle. So Clostridium botulinum produces a neurotoxin. So if you ingest the bacteria, the spores, then you will experience the effects from the neurotoxin. And this neurotoxin, the effects start from the head and work their way down. So the first symptoms are typically blurry vision or double vision. The muscles in the eyes have become paralyzed to where they cannot focus. The eyelids will start to droop. And so this kind of paralysis is called flaccid, because you're not able to get a muscle contraction anymore. They're stuck in the relaxed state. Okay, so then you'll start to have slurred speech, difficulty swallowing, dry mouth, muscle weakness, right? All the effects of not being able to properly contract your muscles. Of course, if that continues to work its way down, then you will paralyze your respiratory system. And at that point, if you're not in a hospital where you can have supportive care, that would kill you, right? You would no longer be breathing. We do have antitoxin available. So if someone comes in with these symptoms, we can administer the antitoxin and stop it from getting to their respiratory system. But also watch them very closely in case it does get to that point and we need to give them a breathing assistance. Now, one of the most common ways that people get this infection is from canned foods that have gone bad, or something broke down in the canning process, because the canning process should kill every and any bacteria, including endospores. But if you look at this example of this can, it's bulging. That means there's gas production happening in there, and the only way to get that gas production would be if bacteria are growing. So if an endospore survived, now it's in this great food, right? In this great condition. It's not crazy hot, it's not crazy cold, it's just room temperature, right? It's just a can. And lots of food to eat. So then it will germinate and start to reproduce as it's that metabolism byproduct. One of the byproducts is gas, right? So that's why this can is bulging. So if you ever see a can that looks like the sides or the top are bulging, like there's gas in it, dispose of it. Do not eat it. Don't try to say, well, I'll just cook it extra long. You cannot kill a protein toxin, right? So toxins, once they're present, that's it. There's no getting rid of it, okay? All right. C. difficile is an interesting bacteria in that a lot of people actually have it as part of their normal gut flora. And so technically it's an opportunistic pathogen. And what happens is the most common reason that people will get this infection is that they're on some type of either long-term antibiotic treatment, frequently on antibiotics, or they've just had a really strong dose of maybe even multiple antibiotics at the same time. And so they've killed off some of their gut flora, and now there's space for the difficile to take over, because you didn't kill the C. difficile with those antibiotics, you killed some of the other flora. And so they're opportunistic. They take that opportunity to grow more space, more food for them, right? But then that leads to all sorts of side effects for you. Having too much C. difficile in your gut can lead to gas and awful diarrhea. And they were not killed by antibiotics, so it's very, very difficult. There are different antibiotics that can sometimes be effective, but it's tricky because you also risk killing other gut flora, and so then you might just have this repeating cycle, right? So this can take a very long time to actually treat and get over. I don't consider it an actual pathogen where if you're not necessarily going to be ingesting this. It is possible if you're around people that have this infection, because they will have such awful diarrhea. It is possible that it will be on surfaces, and that you could potentially ingest it and get an infection that way. But really the vast majority is this opportunistic pathway that I described. Okay, Clostridium perfringens. This is it can cause a skin infection if you have a wound and the endospore gets into the wound. If you ingest the endospore, then it would cause a food poisoning reaction. The food poisoning reaction is actually not so bad. You just have about 24 hours of vomiting, diarrhea, whatever, and then you recover. However, gas gangrene, uh, if it actually gets to the point where you have the symptoms of it's decaying your skin, and you get that bubble of gas forming right below as it's breaking down your tissue, then you have to have typically an amputation to stop it from doing that to any more of your body. Um, Sometimes, like I said, sometimes we can treat it quickly enough, but it's a really awful awful thing. I didn't put images of it. Um, they can be quite disturbing, although I'm sure I have plenty of images throughout this class that are still going to be disturbing to people, but I try not to be overly gruesome. But if you're curious, you could definitely look. Your textbook might even have a picture, I can't recall, but you could definitely Google it. Clostridium perfringens or gas gangrene, and you can see for yourself. The gas is, again, due to the metabolism of the bacteria as it's breaking down the tissue below the skin, and typically the skin is also black. Okay, now tetanus. Clostridium tetani, we have no cure for it. There's no treatment. It's almost always deadly. Okay. We only prevention is right. We have the vaccine. We can prevent it. So you should be getting your tetanus shot, I believe, every 10 years. And if you do think that you've been, a wound, like you were outside and something that's been sitting outside, you get a cut from it, you can definitely go to the doctor and get another tetanus shot just to make sure that you are covered. Okay. You can also have them clean the wound out for you if it's a deep enough wound. Why not have them also clean it out for you? Make sure you don't get any type of infection. Because remember, these endospores, you cannot see them. They're microscopic. Okay, so you don't know if they're present or not. We can assume in this can that right, because we can see the effect, but otherwise you don't know. You can't see them. Okay. Did I say everything I wanted to say? Yes. Okay. So a few more gram-positive examples. We've talked about Staphylococcus off and on in this class. 30% of people are actually what we call carriers of Staphylococcus aureus typically in their nose. It may or may not be a resistant strain. Okay, if they've had antibiotics, it is definitely possible that they could have one of the resistant strains. So MRSA, remember, is methicillin resistant, and that's a whole class or category of antibiotics. VRSA is vancomycin intermediate resistance, and then VRSA is complete resistance, so vancomycin resistant Staphylococcus aureus. And I know we taught you guys the correct way to write bacteria is to have it be in italics, and you can abbreviate the genus name but not the species name, and then we have these abbreviations here, SA, and that's mostly because we're dealing with medical situations where we have to use things that are, I guess, a little easier for different different people within the medical field to talk to each other, but also then interfacing with the public, right? So basically, medicine and public don't necessarily follow the rules of science and microbiology, right? Okay. Now, Bacillus anthracis you learned about already from lab 1. You could definitely go back and review your notes from that lab. Remember, primarily is a respiratory infection when you inhale those endospores. But if you were to ingest it, it could give you some gastroenteritis. As far as I know, it's just your normal run-of-the-mill gastroenteritis, nothing too particular about it. Cutaneous, it can, if you get it in a wound, it can infect a wound, but also nothing really of note for that. It's really the respiratory infection why we talk about Bacillus anthracis, because it can cause an acute respiratory distress, and it can definitely be fatal, right? So remember, with endospores, they're everywhere in the soil and in nature, and you cannot see them, right? Cattle can get anthrax easily because they're constantly in contact with the soil, right? Okay. And of course, anybody with a chronic illness is going to be more susceptible to Staphylococcus aureus. I think I've mentioned that a lot. And Staphylococcus aureus can get into, once it gets into a place, it can pretty much infect anywhere, right? So you can have it in your blood, you can have it in your lungs, you can have it in your heart, right? And in some very severe cases, when it becomes it's in the blood and it's systemic, it's disseminating everywhere, it can even infect your bone, although that's definitely not a common infection, because you would be so severely ill, you'd be receiving treatment hopefully before it ever got to endocarditis and bone, right? Okay. Lactobacillus you are familiar with for good healthy gut bacteria. It's in your yogurt. If you have probiotics, you probably see it on your probiotic. And there's also Listeria. Listeria is actually a pretty scary foodborne pathogen. It can become very severe and lead to all sorts of devastating outcomes, right? So the stiff neck, headache, fever sure sounds a lot like meningitis, right? Confusion, loss of balance, convulsions. If you're pregnant, it can cause you to have a miscarriage, or stillborn, which is got to be one of the saddest outcomes that could possibly happen from a pregnancy, even worse than miscarriage, which is also very sad, and also a very normal part of pregnancy. I don't have the statistics in front of me, but most women will experience at least one miscarriage in their lifetime if they're trying to become pregnant. Of course, if you're on birth control, then that's no longer a risk for you. Premature birth is also very dangerous. Babies are not necessarily completely done developing, and they can have a lot of difficulties. So premature birth is also dangerous. So Listeria can cause any and all of those outcomes if you're pregnant. So, when you're pregnant, you need to take extra precautions to wash all your fruits and vegetables, dry them with a paper towel, cook all your foods thoroughly, right? And any symptoms, call the doctor, find out if you need to come in, that kind of thing. Okay, so of all of the gastroenteritis that we've studied up to this point, Listeria is the only one that I find to be actually frightening. Okay, so we're going to stop here because I want to check that the video recording is working. And then I'll post another, probably 2 videos. All right. Keep up the good work, guys.